Hi guys, welcome back to my channel. This is Dr. Rajeshwar from yrpharmatube. In the previous video, we learnt the oxidation and types of oxidation reactions of phase 1 drug metabolism. If you did not watch the video, hit the I button on top right of this video to watch it. To watch the other topics of drug metabolism and medicinal chemistry, click on the links given in the description right below this video. In this lecture, we shall try to understand the first type of phase 1 reactions, that is, the oxidation reactions catalyzed by cytochrome P450 enzyme, in particular the aromatic and aliphatic hydroxylation reactions. At the end of this lecture, you should be able to describe the cytochrome P450 enzyme, its structure, classification, related hydroxylation enzymes, the mechanism of action, and the activity of cytochrome P450 enzyme in humans. And lastly, to characterize the oxidation reactions, particularly the aromatic and aliphatic hydroxylations with their full chemical equation, cofactors involved, how any drug may be metabolized by hydroxylation, and discuss the relationship between endogenous metabolism and drug metabolism by suitable examples. Cytochrome P450 enzymes are so named because they are bound to membranes within a cell called cyto and contain a heme pigment, that is chrome and P. Cytochrome P450 enzymes, sometimes abbreviated as capital letters CYP, and they are a superfamily of enzymes containing heme as a cofactor that mostly but not exclusively function as monooxygenases. In mammals, these enzymes oxidize steroids, fatty acids, xenobiotics, and participate in many biosynthetic processes. By hydroxylation, cytochrome P450 enzymes convert xenobiotics into hydrophilic derivatives which are more readily excreted. In general, P450s are the terminal oxidase enzymes in electron transfer chains, broadly categorized as P450 containing systems. The term P450 is derived from the spectrophotometric peak at the wavelength of the absorption maximum of the enzyme at 450 nanometers when it is in the reduced state and complexed with carbon monoxide. Most P450s require a protein partner to deliver one or more electrons to reduce the iron and eventually molecular oxygen. Classification of P450s. Based on the nature of the electron transfer proteins, P450s can be classified into 6 groups. The different type of enzyme systems and their functions have been given in the table here. The most common reaction catalyzed by cytochrome P450 is a monooxygenation reaction, that is, insertion of one of the oxygen atom into the aliphatic position of an organic substrate, that is RH, while the other oxygen atom is reduced to water. Many hydroxylation reactions, that is insertion of hydroxyl group, use cytochrome P450 enzymes, but many other hydroxylases exist. Alpha-ketoglutarate dependent hydroxylases also rely on an iron oxide intermediate but lack heme. Methane monooxygenase, which converts methane to methanol, are non-heme iron and iron-copper based enzymes. The structure of cytochrome P450. The active site of cytochrome P450 contains a heme iron center. The iron is tethered to a protein via a cysteine thiolate ligand. The cysteine and several flanking residues are highly conserved in known P450s. They are composed of an electron transfer chain consisting of 3 components, namely a heme protein known as cytochrome P450, a flavoprotein known as cytochrome P450 reductase, and a heat-stable lipid component. A heme protein is actually a family of enzymes. It is a terminal oxidase and its function is to transfer an oxygen atom to the drug molecule, that is RH, and convert it to its metabolite ROH. The second enzyme, the flavoprotein, which is NADPH dependent. It functions as an electron transfer carrier, catalyzing the reduction of cytochrome P450 to the ferrous form by transferring an electron from NADPH. The lipid component is also known as phosphatidylcholine, and its function is to facilitate electron transfer from NADPH to cytochrome P450. Magnesium ions are also required for optimal or maximal activity of mixed function oxidases. The mechanism of these enzymes may involve a series of steps, each step being controlled by an appropriate enzyme system. As discussed in the previous video, these steps can be either oxidative or reductive in nature. For example, oxidation of aliphatic CH bonds involving the cytochrome P450 family is believed to take place by a series of interrelated steps. These steps are catalyzed by cytochrome P450 enzyme, cytochrome P450 reductase, and cytochrome B3 reductase with either NADPH or NADH as coenzyme. See the reactions shown here. Cytochrome P450 is a heme protein molecule in which the heme residue is protoporphyrin 9 containing coordinated iron. When the system is at rest, the iron is in the fully oxidized ferric state. It has been proposed that the substrate initially binds to the active site. This is followed by a reduction of the ferric ions of cytochrome P450 to the ferrous ions state by a 1 electron transfer catalyzed by cytochrome P450 reductase enzyme. At this point, molecular oxygen binds to the ferrous ions and is converted by a series of steps to an activated oxygen species. This species reacts to form the appropriate product. The complete system is non-specific, catalyzing the oxidation of a wide variety of substrates. This is probably due to both the non-selectivity of the enzyme and also the existence of numerous isoenzymes. Furthermore, lipid-soluble xenobiotics are good substrates for the cytochrome P450 monooxygenases because high concentrations of the enzyme system are found in lipoidal tissue. Drugs interact with the cytochrome P450 system in several ways. As discussed before, there are a variety of these enzymes in humans and they are capable of metabolizing a chemically diverse range of substrates ranging from drugs and food toxins to environmental chemicals. Because they are diverse in nature, individuals vary in the types of cytochrome P450 enzymes they may have. As a result, the rate at which specific drugs are metabolized can vary from patient to patient, thus introducing an element of uncertainty into what the therapeutic dose of those drugs should be. Another problem associated with the cytochrome P450 enzymes is the fact that some drugs can affect their activity. For example, phenobarbital enhances the activity of cytochrome P450 enzymes while the anti-ulcer agent cimetidine inhibits activity. This can be a serious problem if the patient is taking another drug, for example warfarin, an anticoagulant, which is normally metabolized by the cytochrome P450 enzyme. Inhibition or enhancement of cytochrome P450 enzyme activity will mean that the blood concentration levels of the warfarin drug will be different from those when the drug is taken on its own. This could lead to subtherapeutic levels of the drug if the activity of the cytochrome P450 enzymes is enhanced, or a drug overdose if the activity is inhibited. Certain foods can also influence the activity of cytochrome P450 enzymes. For example, Brussels sprouts and grapefruit juice, leading to the same types of problems. For this reason, many current medicinal chemistry projects aim to design drugs that are not metabolized by cytochrome P450 enzymes and which have no effect on cytochrome P450 activity. Aromatic hydroxylation or oxidation of aromatic carbon atoms. This is a very common reaction and a major route of metabolism for the drugs and xenobiotics containing an aromatic or phenyl ring. Aromatic hydroxylation is an oxidation reaction in which an oxygen atom is inserted into an aromatic carbon-hydrogen bond. See the equation given. It is to note that the substituent present on the aromatic ring influences the ease of hydroxylation. Thus, it is also called the oxidation of aromatic carbon atoms. Chemically, the hydroxylation reaction is mostly mediated by catalysis and heat. Hydroxylation of aromatic ring by the cytochrome P450 enzyme leads to the formation of a highly reactive strained arene oxide or epoxide intermediate. This epoxide, in the presence of an acid, undergoes hydrolysis to a diol, which means that the epoxide reacts with water and opens to a diol. This is then dehydrated to form an aromatic ring of a phenol. Alternatively, a rearrangement may occur with the formation of a ketone and thence by enolization a phenol. The rearrangement of a hydrogen atom implicit in this process is known as the NIH shift. NIH stands for National Institute of Health USA, where it was discovered. It is to note that in most of the cases, aromatic hydroxylation occurs at para position. In the reaction, the nucleophile in the cleavage of the epoxide may be the nitrogen of a nucleic acid base or the side chain amino group of a protein. The effect of this new bond is to attach the drug to the nucleic acid or protein. This can lead to liver damage, that is hepatotoxicity. The nucleophile may also be the thiol of glutathione. This can lead to deactivation of a toxic metabolite. Nucleic acid bases such as guanosine react with the epoxide ring. Thus, the products are typically phenolic compounds, and the position of hydroxylation can be activated or deactivated by the type of substituents on the ring, according to the theories of aromatic electrophilic substitution. For example, electron-donating substituents on the aromatic ring leads to ortho and para-hydroxylation, whereas electron-withdrawing substituents reduce or prevent ortho or para-hydroxylation. For mono-substituted benzene compounds, para-hydroxylation frequently predominates, with some ortho product also formed. Ropivacaine, a local anesthetic, contains an electron-rich aromatic ring that readily oxidizes to form a mixture of the isomeric 3 and 4-hydroxy metabolites. The newly introduced hydroxyl groups are commonly subject to phase 2 metabolism, which will be addressed in the coming lectures. Moreover, steric factors must also be considered since oxidation occurs mostly at the least hindered position. When more than one phenyl ring is present, only one ring is typically hydroxylated. In other words, in drugs with 2 or more aromatic rings, hydroxylation occurs preferentially in the more electron-rich ring. For example, phenytoin contains 2 phenyl rings, out of which only one ring is hydroxylated to form its hydroxylated derivative. So as diazepam and chlorpromazine are the 2 drugs that preferentially undergo hydroxylation in the electron ring. The benzylic position adjacent to the aromatic ring is also hydroxylated quite easily. An alkene may be epoxidized by the same process as exemplified by the metabolism of the tricyclic antidepressant carbamazepine to its epoxide. A combination of benzylic hydroxylation and epoxidation can be found in the metabolism of safrole to the toxic hydroxy epoxide. There are many examples of aromatic hydroxylation reaction. In the first example, acetonilide is converted to paracetamol, that is 4-hydroxyacetonilide, by the cytochrome P450 liver enzyme. Acetonilide is pharmacologically a weak painkiller, that is analgesic, but the product paracetamol is a more powerful analgesic. In the second example, the aromatic hydrocarbon benzopyrene is converted to its 3,4-epoxy 7,8-dihydroxybenzopyrene, which is a carcinogenic agent. In the third example, the local anesthetic and antidysrhythmic drug lignocaine is converted to its 3-hydroxy derivative. Similarly, amphetamine and fenformin are converted to its 4-hydroxy derivatives. Clonidine, an antihypertensive agent, undergoes little aromatic hydroxylation as the ring is deactivated due to the presence of 2 chlorine atoms. Probenecid, a uricosuric acid, does not undergo aromatic hydroxylation as the aromatic ring is deactivated. Other drugs that undergo aromatic hydroxylation include phenylbutazone, an anti-inflammatory agent, propranolol, a beta blocker, phenobarbital, a sedative drug, fenformin, antibiotic agent, 17-alpha ethinyl estradiol, which is an oral contraceptive, S-warfarin, an anticoagulant, phenytoin, an anti-epileptic agent, amphetamine, CNS stimulant, etc. Coming to aliphatic hydroxylation. Aliphatic hydroxylation is the insertion of an oxygen atom into an aliphatic carbon-hydrogen bond. See the equation. The hydroxylation of an aliphatic or alkyl carbon atoms, that is CH2, can also be catalyzed by cytochrome P450 enzyme. This oxidation can take place either at the terminal methyl group or at the penultimate, that is next to the last carbon atom as shown in the picture. The oxidation at the terminal methyl group is referred to as omega oxidation, and the oxidation of the penultimate carbon atom is called the omega-1 oxidation. The initial alcohol metabolites formed from these enzymatic omega and omega-1 oxidations are susceptible to further oxidation to yield aldehyde, ketones, or carboxylic acids. Unless specifically catalyzed by an enzyme, omega-1 oxidation tends to occur more frequently. Alternatively, the alcohol metabolites may undergo glucuronide conjugation. Aliphatic omega and omega-1 oxidations commonly take place in drug molecules having straight or branched alkyl chains. For example, pentobarbital is oxidized or hydroxylated at omega and omega-1 carbon atoms of its pentyl side chain to form its corresponding hydroxy derivatives. In the second example, valproic acid, which is an anti-epileptic agent, undergoes both omega and omega-1 oxidation to the 5-hydroxy and 4-hydroxy metabolites respectively. Further oxidation of the 5-hydroxy metabolite yields 2N-propyl glutaric acid. See the reactions. In drugs containing a cyclohexyl ring, enzymatic alicyclic hydroxylation generally occurs at C3 or C4 and can lead to cis and trans isomers. For example, acetohexamide, an oral hypoglycemic agent, undergoes alicyclic hydroxylation to give trans 4-hydroxy acetohexamide as a major metabolite. In phencyclidine and minoxidil, it appears that Alicyclic hydroxylation of a six-membered piperidine moiety may parallel closely the hydroxylation of cyclohexyl moiety. Other drugs that undergo omega and omega-1 oxidations include barbiturates, meprobamate, glutathione, ethosuximide, phenylbutazone, ibuprofen, etc. This is the list of references followed for the lecture. That's all in this video. The oxidations catalyzed by cytochrome P450 enzyme in aromatic and aliphatic drugs. In the next lecture, we will discuss about the other oxidation reactions. Till then, never stop learning and never stop watching my videos. Thank you for watching this video.